All right, in the last video recording we were looking at, in addition to looking at how axons regenerate in the peripheral nervous system, we were talking about myelinated versus unmyelinated axons with regard to impulse transmission. And so we were looking at sort of how those being myelinated versus unmyelinated affected the rate and efficiency of impulse transmission down a single axon. But once the impulse reaches the axon terminals, then the impulse can then basically be transmitted to the next cell in line at something called a synapse. So a synapse is where an axon transmits an impulse to another cell, which could be another neuron, it could be a muscle cell or fiber, or it could be a gland cell. And so we refer to that chain, we're looking at a chain of neurons or a chain of cells. The one occurring before the synapse is presynaptic. The cell that occurs after the synapse is the postsynaptic cell or neuron. And so we want to remind ourselves that that the synapses can occur between axons and any portion of another neuron except regions that are myelinated, and asking, why could an impulse not be transmitted into a part of an axon that's myelinated? And that's, remember, because the myelination is generated by the glial cell, and glial cells are non-excitable. All right, so we're looking at a diagram here showing us a presynaptic neuron. So you're looking here at a what kind of structural classification of this neuron would we say this is? It would be a multipolar neuron because it has multiple processes. And what are the characteristic functional types of neurons that can be multipolar? Remember, they are either motor neurons or interneurons. Here we're looking at, we're assuming this is a motor neuron. But in this case, the impulse is traveling down the axon. It's not showing the myelination here. But that would be the presynaptic neuron. Here's where the synapse occurs. It's where the impulse is going to jump over to the next neuron, in this case, in the chain. So this would be the postsynaptic neuron. But this, again, could be, say, a muscle fiber, a skeletal muscle fiber, if the synapse is occurring at a neuromuscular junction, or it could be a gland cell. If say, a sweat gland is receiving an impulse to produce sweat. The pre- and postsynaptic neurons and the synapse can be simplified in diagram here, where a synapse is represented by sort of this Y-shaped ending of the axon and the spherical shaped cell body of the next neuron. All right, let's see. So again, the presynaptic neuron is sending the impulse through the axon. The postsynaptic neuron is then sending the impulse through dendrites and through the cell body into the next part of the cell, which would be the axon. All right. Oops, back up for a sec. This is a transmission electron micrograph of a presynaptic cell, in this case a synaptic knob of an axon, and a postsynaptic cell here. So the synapse is occurring right across this portion here, and this is demonstrating, you can see synaptic vesicles here. It's demonstrating a type of synapse that we'll talk about here in just a minute called a chemical synapse. But there are two different types of synapses, and we'll start with the first type, which is an electrical synapse. So electrical synapses are less common in mammals. They are important where a synapse, or let's say the nerve impulse transmission, has to be very rapid and well coordinated so that the innervation is synchronous across multiple cells at the same time. And that becomes important, say, with cardiac muscle tissue. In the heart, cardiac muscle fibers are contracting simultaneously so that the entire set of ventricles, both sets of ventricles, contract together at the same time. If we have some cardiac muscle fibers contracting at a different time than others, then we have sort of asynchronous contractions, and then the heart doesn't act as a very efficient pump. All right, so electrical synapses allow for rapid and synchronous transmission of impulses from one cardiac muscle fiber to the next through these intercalated discs. Also, this is important between smooth muscle fibers. So that, say we're looking at a sphincter muscle or muscle cells, smooth muscle cells within the wall of a digestive system organ, those muscle fibers need to contract at the same time and efficiently and rapidly to be effective. All right, so here's what an electrical synapse, basically how it functions. Here's our presynaptic cell, and over here's our postsynaptic cell. And this is the plasma membrane of the postsynaptic cell, the plasma membrane of the presynaptic cell, with that gap junction between them. And the gap junction is connected here by these proteins called connexons, these transmembrane proteins, which allow channels through which sodium ions can very efficiently and rapidly pass from the presynaptic cell into the postsynaptic cell. And remember, it's these positively charged sodium ions that are creating a change in the voltage, the electric voltage across the membranes, which is the impulse itself. All right, so this would be how, this is basically the structure and how an electric synapse works. All right, whoops. The next type of synapse, the second type we're going to talk about, is the chemical synapse. And this is the type of synapse that we talked about with regard to innervating muscle fibers, skeletal muscle fibers at neuromuscular junctions. So this is a much more common type of synapse in humans. It occurs between most neurons and between all neurons and effectors. And it involves a neurotransmitter, such as acetylcholine. So a neurotransmitter is a signaling molecule or chemical signal, and there are different kinds, there are other kinds of neurotransmitters besides acetylcholine, such as epinephrine and norepinephrine, which signal our fight or flight portion of the autonomic nervous system. Serotonin, dopamine, GABA, these are all different examples of neurotransmitters. But acetylcholine is the most common type, and we've talked about it before. So here's our diagram demonstrating chemical synapse. So you're looking at the axon terminal. Here's the synaptic knob with these synaptic vesicles containing acetylcholine. As you recall, in chapter 10 we talked about this, where the synaptic vesicle is basically a secretory vesicle that through exocytosis releases acetylcholine into the synaptic cleft. Then the acetylcholine diffuses across the synaptic cleft to bind to a receptor molecule in the postsynaptic cell, or in this case it's showing a postsynaptic neuron. So here's the presynaptic neuron, here's the postsynaptic neuron. Once acetylcholine binds to the receptor molecule, that opens the ion gates so that the sodium ions can enter the postsynaptic neuron, can cross the membrane. And that's going to create a change in the electrical voltage across the membrane, which is, again, the nature of the impulse. Let's see. So information always flows in one direction from presynaptic neuron to postsynaptic neuron. It never flows in the opposite direction there. Their names would just simply change if that were the case. There are two factors that influence the rate of conduction. First of all, it's the axon's diameter, which we talked about earlier. The myelinated axons tend to have a larger diameter, which allows their impulse to transmit more quickly. And then the presence or absence of the myelin sheath. This synaptic cleft, sort of the rate of diffusion is affected by the synaptic cleft, or the distance here between these two cells. So that's what we refer to when we talk about synaptic delay, is how quickly these molecules diffuse across the synaptic cleft. And of course, if there are multiple neurons along a chain, or if we're looking at a reflex where a sensory neuron is synapsing with an interneuron, which is then synapsing with a motor neuron, then the greater the number of synapses, the longer the synaptic delay. All right, and in that case, we're looking here again at those chemical synapses. So that pretty much covers what we're going to talk about with regard to synapses. We have a couple of clinical applications here. One in terms of tumors in the central nervous system. Tumors basically occur not from neurons, but from glial cells and on meninges, which are these protective layers that surround the brain and spinal cord. And so the question is why? Why don't tumors occur within neurons? And if you recall, cancerous tumors are basically cells that are reproducing at an uncontrollable rate. And since neurons are not mitotically active, they're less inclined to develop cancerous tumors. Whereas glial cells, which are mitotically active, can produce gliomas, which you can see here is an example of a brain tumor. And because of the blood-brain barrier, limiting the large molecular weight structures that molecules that can cross over capillary cell membranes and into brain tissue, chemotherapy is not effective in treating those brain tumors. So they either have to be surgically removed or receive radiation. All right, so another application we can look at is some nervous system disorders. On the left, you might recognize Stephen Hawking, who is a famous physicist that in his dissertation essentially provided a great amount of evidence supporting the Big Bang theory. But he suffers from amyotrophic lateral sclerosis, or ALS, which is Lou Gehrig's disease is the sort of common term for it. And it is a disease of motor neurons in the brain and spinal cord that leads to degeneration of the motor nervous system. So someone with ALS syndrome or ALS disease has weakened and atrophied muscles, which you can see causes his sort of slumping in the chair here, but it has no effect on his sensory abilities and cognition. So, so that's why he's been able to continue to be very productive, and as a result of his disorder and his continued productivity professionally and scientifically, there's been a lot of interesting and effective, helpful technology that's been developed to help him continue to function. This is an ultimately fatal disease, but he sort of beat the odds here with living into his 70s, and he developed this disorder when he was in his, I think, early 20s, sometime in college or graduate school. On the right here we have an individual who has multiple sclerosis, which is an autoimmune disorder in the central nervous system resulting from the destruction of the oligodendrocytes. So basically we have an unraveling or dismantling of the myelination of neurons in the central nervous system. And so it affects both sensory and motor coordination and leads to scarring and inflammation that can cause permanent loss of function. Now these individuals can have periods of remission, and can still be functional with the disease over a long period of time. So these are some examples of some nervous system disorders. It's a very interesting area overall to look at disorders of the nervous system, but these are what we'll kind of use as our examples in this class. So that kind of takes us through the material that we were going to add to Chapter 14, as well as to review material that we've already covered for Module 3. So that concludes our video recordings for this chapter. So this was the end of Part 4.